So let me know. Let me know when I could start.
Go right ahead and start.
Yeah. Okay. Okay. So good afternoon, everyone, and thank you for being here. It's a pleasure to have the opportunity to present my research on alternative reproductive tactics in bluegill sunfish. So all organisms must allocate limited resource among survival, growth, and reproduction, resulting in the history trade-offs. While survival is foundational to fitness, it's often tightly linked to growth, particularly in species like fish that exhibit indeterminate growth. In this species, individuals continue growing throughout their lives, and a larger body size often translates to significant advantage in reproductive success and survival. However, allocating resources to growth, delay reproduction, potentially increase the risk of dying before reproduction. Across species, life history strategies often align with the R at K selection continuum, where our strategies prioritize early reproduction and a high offspring quantity in unstable environments, while K strategies invest in growth, delayed reproduction, and offspring quality in stable environments. This trade-off between growth and reproduction is not only evident across species, but also within species, manifesting as distinct reproductive strategies tailored to specific ecological and social contexts. That is the ART or ARTS, alternative reproductive tactics. So ARTS refers to discontinuous behavior and other traits selected to maximize fitness in two or more alternative ways in the context of intraspecific and intrasexual reproduction competition. Individuals use evolved decision rules to allocate resource to one or another way to achieve the same functional goal. Here we have a fantastic study system, the bluegill. There are three male reproductive techniques. The parental male mature at around 7 years old. These males construct nests, herd female, and provide care to young. Males in the parasitic life history became reproductively mature at around 2 years of age. Initially, these males use the sneaking technique to dart in and out of the nest within the colony to steal fertilization while parental males and females are spawning. As they grow up, sneakers transition into a satellite technique and take on female-like coloration and behavior. The last male uses female mimicry to enter a parental male's nest and steal fertilization. These parental and parasitic life histories are fixed once a male adopts one, and they remain in that trajectory. So this image clearly highlights the size difference among arts in bluegill sunfish. And it's important to remember that these fish don't exist in isolation. They thrive in water where their environment play a critical role. This is what they look like in the nest. The variation in how resources are allocated across growth and reproduction is influenced by both intrinsic factors, such as genetic and physiology, and extrinsic factors, including predation risk, food availability, etc. So current research has largely focused on single population studies, and existing ecological studies have investigated association between environmental factors and reproductive behaviors or traits, but seldom assess their effects on ART frequency itself. Consequently, despite evidence that ART frequency vary markedly among nature populations, this remains a critical gap in understanding how specific ecological factors influence the distribution of reproductive tactics at a population level. So our objective here is to explore the ecological drivers and the evolutionary implications of the ratio between parental and parasitic tactics in male bluegill across different lake populations. So first, we aim to examine whether there is a consistent size-based distinction between tactic types across population. And second, we want to determine whether the proportion of parasitic versus parental males vary among populations. And lastly, we aim to identify the ecological factors associated with population level variation in parasitic to parental ratios. So we sampled bluegill from a total of 25 lakes, 24 from Wisconsin and 1 in Indiana. These lakes were selected to encompass broad gradients in size, nutrition level, and the dissolved organic carbon dioxide. We sampled the lakes a few years ago and measured their length and sex. The sex of mature individuals were determined by gently applying pressure to the abdomen to check for the presence of milk or eggs. And if nothing came out, it is considered as an immature fish. We sampled a total of 7,945 bluegills, and the length of captured mature male was distinctly bimodal, as shown in the blue fig here, with 2 prominent peaks. This bimodality reflects the presence of arts in male bluegills. The smaller are the sneakers, and the larger are the parent males. And statuless males were not distinguished in this analysis. We then determined the cutoff separating parental males from sneakers using the kernel density estimation across all lakes. And the global cutoff across all lakes is 120 and 120.9 millimeters. So next, we examine if the proportion of parasitic males differs among lake population. So this is a figure of all lakes of all the males length distribution. We use a global cutoff as shown by the red line, and the ratio is calculated as the number of sneakers divided by the number of parent males. The mean ratio is 1.53, meaning that on average there were 1.53 sneaker males for every parental males. And however, this ratio ranged from 0.01 to 6.36. And our generalized linear mixed model confirms substantial amount lake population with an ICC of 0.5, indicating that approximately half of the variance in reproductive tactics was explained by lake identity. We next want to explore ecological variables linked to the ratio of parasitic to parental males. So to do this, we investigate 5 ecological and population level parameters and formulated the hypothesis for their effects on the ratio. So first, the male density. We hypothesize that higher male density promote parasitic tactic by intensifying reproductive population. In dense colonies, parental males face greater interference and sperm competition, reducing their ability to defend territories and monopolize fertilization. As a result, sneaking or mimicking gain a reproductive advantage, increasing their frequency in the population. Our sex ratio is kind of similar. Male-based sex ratio could mean more absolute number of parasitic males and increased competition for access to female, may reduce the success of territorial male by parental males and favor parasitic tactics, therefore. And next, we hypothesize that high predation pressure increases the frequency of parasitic tactic in bluegill by simultaneously selecting for early maturation and low behavior exposure during reproduction. Next, the water clarity. So fish are highly dependent on visual perception in their foraging and defense behavior. Bluegill, in particular, is a visually oriented species that rely on vision for reproductive interaction. In lakes with high dissolved organic carbon, the dioxide concentration, reduced water clarity may benefit parasitic male by making it more difficult for parental male to detect and distinguish them during reproductive activities, providing sneakers and settlers with more opportunity to exploit parental effort. As a result, higher dioxide levels are predicted to increase the success and the relative frequency of parasitic tactic in such environment. And last, the chlorophyll. The chlorophyll has a similar situation to dioxide as their effect in water clarity, but they can also as an indicator of the productivity. So in resource high in resource-rich environments, more individuals can meet the energetic requirements for reproduction at smaller body size, and the ceratopsian males can begin reproducing earlier, since this might also favor this tactic. So we sampled predator numbers, DOC, chlorophyll, and calculated the sex ratio and the bluegill density, the predator density, and generated this summary characteristic across study lakes. We conducted 2 linear regression models. First is a linear model including all 5 main effects. The second is a full model that included all 5 main effects as long as all possible two-way interactions. So after model selection, the sex ratio was not retained for the first model. The first model explained around 34% of the variation in the response. And in the second model, including all the two-way interactions, the best supported model consists of predator, DOC, sex ratio, and the interaction between DOC and the sex ratio. This model expands around 44% of the variation and with a lower ASA. So initially, when consider only the main effects, predator density and the chlorophyll A concentration emerged as significant predators. However, incorporating interaction terms revealed that although predator density became marginally significant, a notable interaction between DOC and the sex ratio significant enhanced the explanatory power of our model. So let's have a look at this interaction. DOC level may significantly affect the ecological pattern of bluegill IRT by changing water transparency. In clear water conditions, which is low DOC, the relationship between the male to female sex ratio and the sneaker to parental male ratio was pronounced. At low male proportions, which is the pink line here, populations exhibit considerably reduced sneaker to parental ratio, such as in limited reproductive success or frequency of sneaker tactics under conditions favoring visual protection and the competitive dominance by parental males. This may reflect the fact that when the absolute number of sneakers is low, parental males can effectively detect and repel sneakers, provided they have sufficient visual clarity. And conversely, at high male bias ratio, which is the blue lines here, the ART ratio was relatively invariant across DOC level. This invariance could arise because high overall male abundance leads to large absolute number of sneakers, potentially overwhelming parental defense, even when visibility is high. Thus, intense male-male competition potentially override the environmental influence of water transparency on reproductive tactic success. And that's it. So our main conclusions here is mainly on 3 parts. The first part is that we show that the male bluegill show a consistent bimodal size distribution across lakes, validating the use of a 120.9 millimeter lens cutoff to distinguish parasite and parasitic tactics in population level studies. And also we show the ART frequency are highly variable among lakes, and environmental variables and the population demographic structure do not act independently, but interactively shape complex ecological outcomes. And that's it. Thank you, everyone. And I'm happy to take any question if I have time.
It looks like we have maybe 30 seconds or 20 seconds for a quick question. If anyone has one, you just raise your hand and we will open the audio for you. Okay. Well, to keep on time, let's move forward. And if anyone has any questions for Xiyuan, I encourage you to ask them through chat. The next speaker is Brittany Velikaneye. Hopefully I pronounced your name correctly. So what we'll do is hopefully we'll give you some kind of indication at 1 minute or 1 minute before the end of your talk that you have 1 minute. Okay, so I'll put a little thing up on my screen, but go right ahead.
Alrighty. Well, thank you all for coming today. My name is Brittany Velikaneye, and today I'll be talking about some of my dissertation work concerning epigenomic changes under elevated pupal and adult temperature in male and female head tissue. So what we know is that global warming is one of the anthropogenic changes that's occurring due to climate change, and this is a major environmental shift going on Earth. And just to highlight this, here's a map of the world showing recent temperature trends from the 1990s to 2022. And what you see is the majority of the Earth are highlighted in those warm colors. So yellow, oranges, and reds, indicating that those parts of the Earth and the majority of the Earth showed increases in temperature over that time period. And what we also know from Crossley et al. In 2021 is that butterflies here in North America are not responding well to this increase in temperature. As temperature increases, they found species abundance decreases. And so we know that phenotypic plasticity is a way that individuals can respond to changing environments. And so just to give you a quick refresher, phenotypic plasticity is the ability of a single genotype to produce many different phenotypes if they're exposed to different environmental conditions. And one great example of this comes from bicyclist butterflies, where butterflies that hatch in the wet season grow up to be adults that have really large, beautiful eye spots on their wings, whereas those individuals that hatch in the dry season grow up to have small eye spots that you can't really see as adults. And so phenotypic plasticity contributes to the phenotypic variation and available for selection and allows individuals to respond to changing environments. And the molecular mechanism that underlines phenotypic plasticity that I've decided to study is epigenetics. And epigenetics are really cool because they can alter gene expression without actually changing DNA sequence. And so there's three major categories of epigenetics. There's histone modifications, which are the additions and subtractions of chemicals to the histone proteins themselves. So those are the proteins that DNA wraps itself around. We also have non-coding RNAs. There's lots of examples of these, but one you may have heard of is small non-coding RNAs. These attach to messenger RNA and modify gene expression post-transcriptionally. And then we also have DNA methylation. And so this is the addition or subtraction of methyl groups directly to the DNA strand. And so what I'm going to be talking about for the rest of the talk today, when I'm speaking about epigenetics, I'll be talking about DNA methylation. And so I just wanted to go over how DNA methylation might work in this hypothetical situation where you have a code for wing color. This particular code codes for a dark green butterfly. If we expose the individual to a changed environment, say an increase of temperature with a heat wave, that individual may be able to respond to the change in its environments by adding a methyl group to somewhere inside this wing color gene. That will allow the individual to change its gene expression of this gene, allowing it to become a light morph butterfly that is much more better suited for this warm environment as it absorbs less heat. Additionally, if we remove the temperature change, so the heat wave ends, the individual is able to once again respond to its changing environments, removing that methyl group, reverting its gene expression, and changing its phenotype back to the dark green butterfly, suggesting that DNA methylation may be also reversible. And if you're coming to me today from a talk or a study system of vertebrates, it's just important to know that diversity in methylation-based gene expression varies among groups. So in invertebrates, where we understand a lot of DNA methylation, we know that methylation occurs most frequently in promoters. And when the methylation occurs in promoters, accessibility to transcription factors is decreased. And because we have that decrease in accessibility, we also have a decrease in expression. However, in invertebrates, like the butterflies and moths that I work with, most frequently methylation occurs within the gene itself, so in its introns or exons. And when methylation occurs there, what we see is increases in transcription factor accessibility. And because we see increases there, we also see increases in gene expression. Additionally, what we know about DNA methylation in insects is that heat exposure increases DNA methylation. And so here are just 4 examples of previous studies done in insects. Each one of these studies found that when they exposed individuals to heat, the overall methylation also increases. We also know that DNA methylation patterns can differ substantially among different tissues. So here is a study by Hunt et al. Published in 2024. They did their study in bumblebees. They found that 134 differentially methylated genes when they compared male sperm to female ovaries. However, when they conducted the same analyses in the brains of these individuals, only 25 genes were differentially methylated. And so this suggests that including tissue-specific analyses may help uncover the extent to which epigenetic regulation is specialized in insects. And then insects also lack consistency in sex differences. So this is kind of where our gap lies. So we know that some species like these mealybugs show female bias methylation, whereas other species show male bias methylation, and others still show no sex differences. But every study that's been conducted in butterflies and moths so far has either done their analyses with the sexes together or only picked one sex. So we don't know exactly where butterflies and moths fall along this continuum. And so that leads you to my question today of how does DNA methylation vary among the sexes in Lepidoptera in response to heat? And so we chose to answer this question in the study system of the European corn borer moth. It is a widespread generalist herbivore, and because it is widespread, it's incredibly well studied and has substantial genomic resources, the resources needed to do epigenomic studies like the one we conducted. And so I just want to highlight some of our previous work that was published earlier this year in Molecular Ecology, where we explored sex differences in methylation in abdominal tissues. And so we explored this in 2 conditions, one where both males and females were exposed to a control temperature, and then the same comparison, but when they both experienced heat. And so here is the results of that study. On the x-axis of that graph, you have the sex of the individual. On the y, you have methylation percent. Females are purple circles and males are represented by green triangles. And what you have on your screen here is when both males and females were exposed to control temperatures. And it's important to note that there's a modest but significant increase in methylation in females compared to their male counterparts. And the same exact pattern holds when both sexes were exposed to heat. Females have higher global methylation compared to their males. And so what we did was we took the one comparison, the heat comparison when males and females were exposed to heat. And here in our current study, we repeated that, except we did it in the heads. And so to do that, we compared DNA from males and females after they've been exposed to heat, sampling DNA only from their heads. We took 12 heads total. We did 4 heads per pool, resulting in 3 female pools, and we did the same exact thing for their male counterparts. Once we extracted the DNA, we used a method called enzymatic methyl-seq to determine where methylation was throughout the genome. This is very similar to bisulfate sequencing, if you're familiar with that, but it's been shown to be less damaging on the DNA. And so how this works is you take the DNA strand, you expose it to an enzymatic treatment. What that does is it converts unmethylated cytosines to uracils, which you can see in that second to last base pair, whereas methylated cytosines remain. Then it undergoes normal sequencing. And what you get out as the sequencing data is in the enzyme-treated sequence, those unmethylated cytosines now look like thymines, whereas methylated cytosines remain cytosines. And by comparing that to a reference genome or untreated sequences, you're able to determine where methylation occurs throughout the genome on a base pair scale. Once we got the DNA sequences back, we put our data through a custom bioinformatics pipeline where we were able to first trim and align the genome. Then we called methylation state and removed single nucleotide polymorphisms using the opposite strand. And then finally, we were able to filter coverage and do 2 different types of analyses. One comparing the overall methylation like you saw we did in the abdomens, and then we also tested for significance of differentially methylated regions, so where those differences between the sexes occurred throughout the genome. And so here on your screen is data that you already saw. This is the data from the abdomens where females have higher methylation compared to their males. The only thing that has shifted is that y-axis now shows a methylation standard. This allows us for a better comparison between the two different types of tissues. And so now I've popped up on the screen the data for the heads. And you'll notice that there's still a sex difference. However, the pattern of the sex difference has completely shifted. Here we see that the heads now have higher DNA methylation in their males compared to their females, suggesting that sex differences in methylation are not consistent across the different tissue types. And so then, like I said, we were able to determine where these differences occurred in what genes. And then we asked which pathways have enriched sex differences. And so the first pathway that we found was the Wnt signaling pathway. This controls body accessing pattern, cell fate, cell proliferation, and cell migration. So it's an incredibly important pathway and is actually necessary for proper formation in insects. Additionally, we found genes that have known associations and known functions with neurogenesis, again suggesting that in the heads of butterflies and moths, perhaps the sex differences lie in the neurogenesis genes, at least on the DNA methylation scale. And so we were able to successfully answer our question of how does DNA methylation vary. We found that males and females differ. However, the pattern that exists between the males and females in those sex differences are highly tissue specific in insects or in butterflies and moths. And combining that with the work of Hunt et al., it suggests that it's also in insects. And so this really suggests that species variability and sex differences could be confounded by the tissue types when making comparisons. So remember in my introduction, I talked about these three categories that species of insects can fall under. And if we just took the abdominal data that we collected, we would say butterflies and moths fall inside the female biased methylation category. Whereas we took just the heads, we would have said butterflies and moths fall in the male bias category. And perhaps if we had sampled in a different section of the body, like the thorax, we would have found no sex differences at all. And so this really suggests that our comparisons across studies may benefit from a more nuanced approach and careful consideration of tissue type, what sexes, and even the environmental conditions those organisms experienced before sampling. And I just want to leave off today with future questions. So one great question would be to answer how methylation is related to changes in gene expression. As I said, there's that general trend of increased methylation equals increased gene expression in insects, but it seems to be more nuanced than that inside each species. So it'd be great to answer those questions in the European corn borer moth to connect changes in DNA methylation, then gene expression, then phenotypic data. And then ultimately also looking to see how sex differences in methylation are ultimately regulated. And so with that, I'd just love to end my talk today by talking or giving acknowledgements to those that helped along the way. So my lab partners for doing all the DNA extractions with me, the Dockman lab at Tufts University, which gave us the European corn borer moths that we used in these experiments, and then obviously the funding that made all of this experiments and all of this research possible. And with that, I'll take any questions if I have time.
Excellent. You have time, so if anyone has a question, please raise your hand. I'll ask a quick question if you don't mind.
Of course.
So that was really fantastic. Tell me, do you think that, or how many other studies across different types of insects have used different tissues? Is your study the first one?
No, so that Hunt et al. Paper also did multiple tissue types. To my knowledge, those are the only two that have started to do that. So I mean, methylation in insects is pretty understudied as it is. There's only 25 studies that have done it in butterflies and moths. So, and we're all doing different looks and takes on it. So I think we're starting to acknowledge how unique and specialized it all is. And so as we grow, I think we'll be able to parse out some patterns, but right now we just haven't got there yet.
Oh, that's fantastic. Thank you so much.
Thank you.
Okay, Louis-Philippe Bateman, you're up. Ready to go? Okay, and I'll give you a note at 1 minute before time, okay?
Sounds perfect. Thank you. Okay, so, well, hi everyone. My name is Louis-Philippe. I'm a master's student at McGill University under the supervision of Professor Hans Larsen. And I'm very excited to be talking about this was my undergraduate honors thesis work. So evolutionary feeding innovations are really important in evolutionary biology. I'm sure all of you know about this, how, for example, fish, mammals, and birds, all to a certain extent, have been able to radiate thanks to their key innovations in their feeding systems. One particular group that did this is dinosaurs, and specifically herbivorous dinosaurs, which evolved herbivory multiple times and in multiple different ways. So across different dinosaur groups, we see all sorts of different muscle arrangements, different cranial bone configurations, different tooth shapes, different masticatory cycles that have permitted herbivory to evolve. What's particularly interesting with herbivorous dinosaurs is that a lot of them evolve or seem to evolve around periods of drastic environmental change. So specifically in around the Middle Cretaceous, floral environments around the world changed quite a bit. So whereas they used to be dominated by ferns, lycophytes, cycads, and conifers, they all of a sudden became dominated by flowering plants, also known as angiosperms. And so previous research has had mixed results as to whether this really influenced dinosaurs, whether in terms of species richness or of biomechanics. So, for example, this study by McLaren et al. In 2016 found no association between functional disparity in hadrosaurs and the abundance of angiosperms. But this other study in 2017 did find that ceratopsian frills were changing as a function of how abundant angiosperms were. So it's still really an open question as to biotic interactions were really affecting the evolution of herbivorous dinosaurs. And so we wanted to weigh on this question by looking at a seldom researched but very, very cool group of dinosaurs, the best group, if you ask me, ceratopsians, which are very well known for their frills, their sociosexual display structures, and also for having been one of the dominant dinosaur groups in the late Mesozoic. So ceratopsians are really interesting as a study system because they preserve 95 million years of feeding evolution. Just to put that into perspective, that means that the latest ceratopsians, such as Triceratops, were further away from the earliest ceratopsians than they were to us. So that gives you some idea as to how long they existed. They're also really interesting because their morphology changed quite a bit over time. So they started off as these very small, like dog-sized animals and evolved into these 6 to 10 ton, twice the size of an African elephant beasts that would probably have had a very important impact on their ecosystems. What's also interesting is that their teeth changed quite a bit. So they went from being these small individual leaf-shaped teeth into complete, very complex batteries that functioned as single slicing edges. Another notable aspect of their morphology is that they developed these very impressive frills and horns and ossifications that they're probably best known for. There's also some tantalizing evidence that something was going on. So this study by Kunz and Sakamoto in 2024 found that relative bite force, which is estimated by looking at cranial proportions, changed quite a bit in ceratopsians to the point that the rate of evolution in this group was actually higher than in other herbivorous and carnivorous dinosaur groups for this specific feeding performance metric. So the specific goal of this project was to understand whether or not angiosperms affected ceratopsian feeding performance. And I do want to point out that we're looking at feeding performance here. So not just morphometric traits or anything else like that might not preserve a very clear signal of how ceratopsians are evolving. And so we have two alternative hypotheses. So if there is significant change in feeding performance, in relevant feeding performance, then it's probably the case that angiosperms did affect the evolution of ceratopsians. And if there's no significant change, well, then it probably didn't respond to the evolution of angiosperms. So what we did is that we went around the country, Canada, and scanned a bunch of ceratopsian skulls, every ceratopsian skull we could get our hands on, actually. I have to thank many colleagues for this, but especially Alex and Tony, who really helped out quite a bit in scanning all these. Canada's a big country, so it took a lot of time. We have a pretty good data set in the end. Most of our specimens obviously are from Western North America, from the late Cretaceous, but we also do have one specimen from the early Cretaceous of China that was scanned and reconstructed as part of a previous study. We also have decent phylogenetic coverage. So we have many ceratopsids, which are the most derived group of ceratopsians. We have one leptoceratopsid, which is another derived but quite different group of ceratopsians. We have one basal ceratopsian and one outgroup here, Pachycephalosaur. In terms of body size, here the skulls scale to the actual body size. So we also have an interesting coverage in terms of body size. Here they are compared to in a more easy way. And so as you can tell, some of these are quite big. So the next part of the project after collecting all these data was to model muscles onto them. So we did this using Blender, which is a 3D modeling software. We based our reconstructions off of studies that have looked at dinosaur muscle anatomy by looking at muscle scars, but also muscle arrangements in dinosaurs and crocodiles to make like phylogenetic inferences as to how they would have attached. There are two major muscle groups, just for some quick anatomy lessons. The pterygonius muscle group, which is involved in sort of crushing bites and sort of stabilizing the jaw as it closes, and the adductor mandibularis externus group, which really is just the main workhorse when it comes to closing the jaw and exerting large amounts of force. We measured 4 feeding performance metrics that we thought would be sort of salient predictors of overall feeding performance in these animals. So bite volume, jaw gape, shear, and bite force. Bite force is particularly interesting because it gives us a good insight into the strength, the toughness of the diet that these animals were feeding on, and also just because it's so widely used and it's been measured in so many taxa that it's a useful comparator. We also measured jaw gape with the assumption that a small jaw gape would result in feeding on smaller food and a larger jaw gape would result in feeding on larger food. This seems to be the case, at least in tropical birds that were measured in this very important study by Wheelwright in 1985. We also measured bite volume, which is less often measured, but we made this assumption that it's probably correlated with selectivity, whereas small animals with small bite volumes, like dukers, are fairly selective feeders, whereas animals like white rhinos, which have very large bite volumes, are much more unselective. We also measured shear pressure, or just pressure, which is just a measure of force divided by area. And the idea is that bite force might not always be the best predictor of how tough a diet is, whereas shear in this case really tells us how much force can be inputted on a specific area. So just quickly like a first interesting factoid from our results is that it turns out Triceratops is the strongest biting herbivore in all of Earth history, at least based on our models. So it has a bite force of 30,000 newtons at the back of the jaw, which is very, very impressive. For comparison, humans have a 300 Newton bite force. So much, much stronger than we are, and also stronger than all these mega herbivores that are around today, such as elephants and rhinos and hippos. In terms of actual evolution, so we took all these values, put them into a phylogeny. Every parent node here is just like an unweighted average, so that there's no complex evolutionary rates. We're still working on that. But what this tells us is that broadly, bite force increases throughout the phylogeny of ceratopsians. Jaw gape actually decreases. So the earliest ceratopsians and this outgroup taxon have a very high jaw gape, but the most derived ceratopsians, such as Triceratops, have a very low jaw gape. Interestingly enough, bite volume drastically increases from being very small, implying selectivity, to very, very big bite volumes. And there's no clear trend for shear. In fact, it's pretty surprising because this very basal taxon over here, Psittacosaurus, has a very high shear value, whereas a very derived Pachyrhinosaurus has a pretty low shear. And we're still working on figuring out why that's the case. Just really quickly, as you can tell, basal taxa are performing very differently than derived taxa. Basal taxa have these like low bite forces, tiny beak volumes, high shear values, and these complex mastication cycles, whereas derived taxa have high bite forces, high bite volumes, average shear values, and these much more simple, like scissor-like masticatory cycles. I do want to quickly nuance this by saying that there are some slightly divergent feeding trends. I won't go over them super precisely, but different families have slightly different feeding performance metrics, implying that there is some sort of niche partitioning going on when they do overlap. But so to get to the meat of the discussion, like, are angiosperms affecting ceratopsian evolution? So to answer this question, we need to know if there is a relevant change in biomechanical properties that's going on during the angiosperm evolution, which takes place about over here in the phylogenetic tree. And so we do see some change, for example, in bite force, and we do see some relevant change. So for example, we would suspect that bite force would increase because angiosperms have tougher tissues than gymnosperms on average. And indeed, bite force does actually increase. And similarly, because early angiosperms had this like weedy, fast-growing habit, we would suspect that ceratopsians would become less selective. And indeed, they do have larger bite volumes, which does imply that they're becoming less selective over time. But we also do have to consider other factors because we're good evolutionary biologists, right? And so it turns out that a lot of these feeding performance metrics just scale very strongly with body size. And if you remember from the beginning of the presentation, ceratopsian body size increases quite a bit through their evolution. Just this here is a graph of bite force over skull length, and it shows you from literally the smallest smallest terrestrial tetrapod alive today to Triceratops. There's this over 4 orders of magnitude. There's a very consistent scaling relationship between bite force, and similar thing going on with bite volume and other metrics. There's also this evolution of very fascinating sociosexual display structures that's going on, which is really modifying the architecture of the skull to the extent that it might be affecting feeding performance. And so when we take these two factors into account, allometry and sociosexual display structures, did angiosperms drive ceratopsian feeding evolution? Well, we don't know. It might be adaptive, it might not be. And so I don't like leaving you guys on a cliffhanger, so I will say we are trying to improve the coverage of our data set by including more species from really during the angiosperm revolution, and we're also going to 3D print some dinosaur jaws and mechanically test them and look at patterns of character evolution to try and really tease out what's going on. Here are my references, and I have so many people to thank, especially lab colleagues, but also everybody who's let us access their museum displays or collections to scan ceratopsians. So there you go. Thank you, everyone.
Exciting, nice work.
Thanks.
Does anyone have questions? Okay, can I ask a naive question as a plant biologist?
Of course.
Do you think did these organisms fought with one another, right? Would there be any, selection for fighting on the skull size and the bite force?
Absolutely. I think that's definitely a good hypothesis. I mean, there's very good evidence for ceratopsians fighting each other from like just a lot of wounds being preserved around like the cheek area and the frill, which does imply some sort of horn to horn combat. And I guess from what we know from modern organisms, having a larger body size definitely does seem to be a big advantage when you're trying to fight off other members of your species. So I think, yeah, sexual selection might be a big driver of all of this. And so even to a certain extent affecting feeding performance. And so that might really just be the explanation for this and not biotic interactions. Yeah.
Nice. Any other questions? Awesome. Well, thank you so much. That was a great talk.
Thank you very much.
Okay, and next up we have Daniel Schönberger.
Can you see my slides and hear me?
I can't see I can see your slides now, and I can hear you.
Perfect. So welcome to my presentation. I'm Daniel Schönberger, and today I want to present you some insights into environmental clustering and how we can use that to test for niche divergence. And niche divergence and testing for niche divergence is quite important in many fields of ecology and evolution. For example, if we want to understand species coexistence or invasive species, but also if we want to study geographic ranges of species, species diversification or ecological speciation. So many different fields, but then we also have to use it in species delimitation. That's my current research approach. And then during the last couple of years, integrative taxonomy has become more popular, where we combine evidence of multiple disciplines, so molecular work, morphological work, environmental data, as in this example here, where they're using climatic data. And then we can use this data to actually infer our taxonomy units and their boundaries in a hopefully meaningful way. There are actually many niche divergence tests now available. The three most common ones are listed here. So the first one, the niche identity and background test, is implemented in mTools, which is quite common, but we also have the niche overlap test, which normally uses Schönberger D. I think that's the most commonly one. And we can also test for hypervolume overlap. So these three different tests, they're actually quite often used. They're really well established. But then if you think about species delimitation, we often have these species variations with so many species. And if we want to test for niche divergence between all of these species, either in pairwise comparison or between closely related species, we need something that is fast, that is simple, and that can implement multivariate data. And these three tests that I've showed you so far, they have some shortcomings for that. For example, the first one, for that we have to implement a niche modeling first, which is quite complicated and takes experience, while the second one only includes 1 or 2 PCs, so it misses a lot of variation, while the first one actually is quite computationally intensive. So we need something fast, simple, and something that can implement this multivariate data. And my approach now is basically doing that. I have 2 different methods that I'm trying out right now. The first one is using environmental clustering and a DAPC. And the second one is a machine learning approach using self-organizing maps. And I'm testing these approaches in this specific study system here, the Meyer BACMOS. And this is really cool because it has 11 taxonomic units that are actually, it seems to be a species variation that is very, very young based on current evidence, probably only a few 100,000 years old. And we have a lot of morphological variation, both between species and within species. So here you can see variation in these wing patterns in the adults, but also different patterns of color and shapes in the larvae. And we also see a lot of ecological variation in terms of what habitat they use and in what host type they use. Previous research hasn't been really successful or not fully successful disentangling the drivers of diversification in this complex. And it's also not entirely clear what are the taxonomic units and the boundaries of these units in the system. There are many library adoptivists or systematics that are people that assign new species based on limited amount of evidence or new subspecies. Therefore, the system is the perfect study system to test my environmental clustering approach. The first one where we combine environmental clustering and a DAPC. And just to explain to you what a DAPC is, it's a really cool method developed 2010. And it's actually mostly used for genetic data, but we can also, as you can see later, apply it to environmental data. And normally it's used for this investigating population structure. So here you can see these 3 different populations that we have. We visualize the first 2 PCs of our genetic data. However, the issue here is that PC summarizes overall variability among individuals. And what we actually want is that to maximize between group variation and minimize within group variation. And that's exactly what this discrete analysis is doing. And you can also see from the scatter plot that it's already well better to group these different populations in a better way next to each other. However, the big issue here is that this discrete analysis can only take a low number of variables that have to be uncorrelated. And normally if you have genetic data or highly multivariate data, we have many variables and they are also correlated with each other. So that's a big issue here. We can't really use this method. However, the solution is that we can use a DAPC. That's a really elegant solution because it takes, well, first conducts this PCA and then retains some of these PCs that include most of the variation in our data and are uncorrelated to each other. And then we can put that in our discrete analysis. And the result is that we can now actually focus on the within group variation and between group variation and separate these clusters in quite a suitable way. So overall, we can also, or what is normally done is combine this method with a clustering approach. So we see how many clusters is in our data, in this case, our genetic data, in this case, these 3 populations. And this is basically a method that is combined. Overall, this method is fast, simple, and can include many variables, so it's suitable for also environmental data. And my aim is to adapt it to this environmental data for species delimitation. So many different species, and then we can test for niche divergence here. And I will show you how this works based on this one species here, Hemiluca nevadensis in Western US. And this species has recently been split into two different species based on a recent preprint and limited amount of evidence based on mitochondrial DNA and pheromone attraction trials. And now I'm testing, are these actually 2 different species or need to be lumped together based on our current evidence? And environmental data facilitates this to give basically another perspective on these 2 species, allowing to test, do they occur in different environment? Have their niches diverged or not? First step is to compile occurrence data. So in this case, we have around 750 unique localities compiled by online databases and databases or records from our collaborators. And then the second step is to compile environmental data or rasters. And I think it's really cool that nowadays we have so many different variables available in really high resolution. And I think it's really important that we not only include this climatic data that is most commonly included, but we also include other abiotic and biotic factors to capture the full spectrum of these abiotic and biotic variables. So in this case, we have a vegetation index, for example, terroir, soil variables that might be relevant for our moths that pupate in the soil, but also matrices that reflect urbanization. We can then do a clustering approach, in this case HD scan, which is quite suitable for this kind of data. And in the end, we do our DAPC on our 2 species. And we can see here, based on this plot, based on one of this discriminant axis, or the only one, we can see that both of these species separate really, really well in this environmental space. And this basically suggests that there seem to be 2 environmental niches, or more or less that both species occur in completely different environment. We can also extract the variables and their relative contributions to the separation. In this case, one variable is really distinctive, namely the number of frost-free days in September. And September is basically the main flying period of the northern individuals. So that seems to be quite important here. And we can also do a permutation test where we actually reshuffle all our species label many, many times, which is basically a null model where we kind of simulate a species, or where we kind of lump both species together. And then we can see, in this case, again, our observed mean is quite different from this null model. This was the first approach. It was fast, it's simple, can integrate this multivariate data. And the second approach is the self-organizing maps approach, implemented in this delim-sum R package. And to explain how it works, so we have this sum is basically an unsupervised neural network that is trained. And what it's doing, it's basically projecting this highly multi-dimensional data that we have into this 2-dimensional grid that you see here. So that's a 2-dimensional map based on these clusters. Each of these clusters is a neuron in our model, and it clusters together individuals or data points based on their similarity. So each of these clusters contains similar individuals, And now we can run a clustering algorithm on that and see how many, or in this case, we can see these two different clusters corresponding to our two different species are well separated. We can do the same thing and do some kind of model comparison based on the output where we see k equals 2, two different environmental niches is well supported based on a much lower BIC value compared to one similar environment. And there are many other tools in this package. For example, we can also do the structure-like plot where we can investigate each occurrence point and see how likely it is that it belongs to one of these two clusters. And the main advantage of this SOM approach is basically that we can integrate not only highly multivariate data, but many different data types. So not only continuous data, such as climatic or soil data, but we can also use categorical or binary data, such as host use or habitat use. And this might be, these are probably two variables that are highly explanatory in our system and might give further insights here. And then finally, we can also see how each of these data layers and variables contribute to our variation in our data. So overall, both of these approaches are highly suitable methods to test for niche divergence or different environments in our study system here. And they're fast, simple, and they can integrate this multivariate data based on my example here. And just as a last word, I want to say that we are currently working on this delim-SOM-R package. It has been developed by Alex Pyron, my collaborator, and now we're working on a second version that is more user-friendly, more accurate. And this is a great tool to do integrative taxonomy, where we integrate multiple data types, so molecular, morphological, and other kind of data, such as this environmental data, to do a fully integrative taxonomy approach. And we'll talk more about it in my in-person talk if you're interested. With that, I want to thank my supervisor, Julian Dupuy, and my collaborator, Alex, and all my funding sources, and I'm happy to take any questions.
Very nice. If you have questions, please raise your hand. I'm not seeing any questions, so I'll ask a simple question. This looks like a really interesting package. Is it available? Is it publicly available yet?
Yeah, it's already, I mean, on GitHub, the code is on GitHub, and we're
Okay.
currently basically working on a more user-friendly version of it, but it's still already available, definitely. Yeah, you can use it, you can use the functions. It's really useful for any kind of multivariate data, basically.
Excellent. Thank you. Okay, I don't see any other questions. And with that, that's actually our last talk of this session. Our final person had to cancel. So I just want to thank all of the speakers who gave talks. These were excellent short talks. You did a great job. And if there's any other questions for the speakers, please feel free to email them those questions. We could open up the chat, or just everyone could ask questions if they want to now. So I'll give maybe like a couple minutes to see if anyone's interested in asking any of the speakers that presented any sort of question.
And in the meantime, Brian, can you share the missing speaker slide just so that anybody who pops in looking for the speaker's talk knows that they're not going to be here? Thanks. I guess we need to update that slide.
Daniel, one more question for you, if you don't mind. What other organisms or systems has your program been used on so far?
So the package, the SOM package has been, I think, used in a lot of salamander studies and other studies in terms of integrative taxonomy. And I'm currently working on this moth system and probably also in papilio butterflies on it. But currently I'm just trying different methodological approaches right now and see how well they are to actually test for niche divergence.
Are there any organisms that you're looking for in particular that could help fill in any gaps or holes?
I think the main point is that we can implement this different kind of data right in my study systems with the bug mouse. It's really cool because we can implement this host use, for example, from we can also implement some kind of maybe pheromone attraction trials, reproductive strategies and all of that. So this different kind of data and put it all into one model. And I think that's the cool thing about the SOM approach, because we can just throw it all into one model and then see what it gives us in the end, basically, because it has this neural network basically allows us to do that.
Nice.
Yeah.
That's great. Well, thanks again. Okay, well, I'm going to go dark, but thank you all for attending this session, and hopefully I'll see you in the in-person meeting this summer.